Welcome to today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Alstrom, and I'll be joined today by my myeloma friends and co-hosts, including Jack Aiello, Gary Peterson, Pat Killingsworth, and Lizzie Smith. This is the sixth in a very important series featuring the Myeloma Crowd Research Initiative. Now, if you haven't already heard about the MCRI, educated patients are teaming up with myeloma researchers to find and fund ideas in myeloma that could have the greatest impact for the next generation of myeloma therapies. We decided to go after high-risk myeloma because these patients have very poor outcomes and very few working therapies. Ultimately, if we become relapsed or refractory to current drugs out there, we also become high risk. And what's been found is what works in high risk will help low or standard risk patients as well. So here's what we've done so far. In February, we asked researchers around the world to submit their proposals, and we received 36 high-quality letters of intent back. In March, that list was then scored by our scientific advisory board, who selected their top 10 proposals. And in April and May, we're now holding Myeloma Crowd Radio shows, so you can be involved. We'd love for you to understand the proposals, so please listen in, ask questions, read the transcript after the show is posted, and share it with your friends and family. This is very critical work being done in myeloma. After the full proposals are submitted and the shows are complete, the Scientific Advisory Board and Myeloma Patient Advisory Board will together decide on a limited number to fund through patient-driven campaigns. And again, we will need your help to get the word out and share the really amazing work that's being done. Now, each myeloma patient has a whole community around them who want to help, but sometimes they don't know how. And this will be a way they can help you find a cure for your disease. So watch out for our upcoming announcement next week on how you can build a team and get started. So we are very privileged to have with us today Dr. Frank Zhan of the University of Iowa, and joining him is Dr. Guido Trico. So welcome, doctors.
Hello, good afternoon. Good afternoon.
Okay, thank you so much. Thank you so much for participating, and let me introduce you both. Dr. Frank Zhan is Professor of Internal Medicine in Hematology, Oncology, and the Blood and Bone Marrow Transplantation at the University of Iowa. Dr. Zhan received his medical degree at Hunan Medical University in China and his PhD in cancer molecular genetics there as well. He performed his postdoctorate work in both Kentucky and at the University of Arkansas for medical science. The long-term objective of Dr. Zhan's lab is to develop new therapies to overcome drug resistance and deplete cancer stem cells in myeloma. His work focuses on finding the genes that make myeloma cells resistant to chemo, and then use that knowledge to test new hypotheses in the lab and in mouse models. Dr. Guido Trico is director of the Bone Marrow Transplant Program at the Holden Cancer Center at the University of Iowa. Dr. Trico was educated in Belgium and has served as the director of the Bone Marrow Transplant and Myeloma Program. Additionally, for Huntsman Cancer Institute, the Greenbaum Cancer Center at the University of Maryland, and at Indiana University. He was also the Director of Clinical Research at the University of Arkansas Myeloma Institute for Research and Therapy, and Dr. Trico has been treating myeloma patients for over 20 years. So again, welcome to you both. I think we will add another one of our co-hosts. Pat, are you on the line?
Hi, Jenny. Can you hear me?
Yep, we can. Thank you for joining. And Jack, thank you for joining us as well, Jack. So I think we would like to get started with some basic biology. So maybe Dr. Trico, you'd like to help us understand what happens when genes are translocated, or what does that mean?
So every gene is normally located on a certain chromosome. When a gene is translocated, it goes from one chromosome to another chromosome. And by doing that, it basically jumps away from where it originally belongs to an area where it thinks it has more benefit to be there than in the original area. That's the basis of a translocation. So it's part of the chromosome that is jumping away from where it used to be to a new chromosome.
Okay, and it seems like the 14 gene is usually involved in myeloma. When you say you have these translocations, there's a 414, a 1416, 1114, a 1420. Why are they all involved in gene 14?
14q, the long arm of chromosome 14, is the place where the heavy chain of the immunoglobulin, and myeloma is a disease where the plasma cells make abnormal M proteins, which are derived from normal immunoglobulins. And immunoglobulin has a heavy chain and a light chain. And the heavy chain gene is on chromosome 14q, on the long arm of chromosome 14. That's why it's so frequently involved in myeloma translocations.
Okay. So today we're talking about specifically the 1416 and the 1420 translocations, correct?
Yes.
And it's my understanding that when a translocation occurs, that it changes certain or very specific genes. Can you explain what genes are affected by these translocations and then what those genes do?
The reason why the important genes go to chromosome 14 is because the promoter of the gene for the heavy chain for myeloma cells is a very strong promoter. And a promoter needs to be seen like an ignition on a motor. The motor by itself doesn't work. Gene is only transcribed when the promoter is active and when it ignites the gene to work. The promoter of the heavy chain gene in myeloma, which makes all the immunoglobulin, is extremely strong. And so what the other genes are doing is they're jumping to that chromosome 14 so that they are now coming under the direction of a much stronger promoter, a much better igniter than where they were originally. And therefore they are way overexpressed in that way. And they have much more activity in that way than if they were at their original place.
So what might have been contained before by regular processes are changed?
Changed, yes. So those genes, they take advantage of the fact that the heavy chain promoter is such a strong promoter, so that they now come under the direction of this very strong promoter, which they didn't have before. And therefore they are much more expressed and much more of that abnormal protein is being made. Okay,
so on the Myeloma Crowd site, what we decided to do is to break up these different Facebook groups into gene-specific Facebook groups. So we have a general group and a family caregiver group, which patients can join if they would like. But we also have gene-specific groups. Can you just give an overall assessment of why the knowledge of your own genetics of myeloma is so critical and how you determine what your features are?
When you look under the microscope, all the myelomas look typically about the same. There's not a lot of variation. But when we looked at gene expression profiles of patients with myeloma, and we have done that on over 400 patients, we saw that there are clearly distinct groups. And the groups are mainly made by the fact that myeloma cells have more than 46 chromosomes, which we call the hyperdiploid group, or have translocations in it involving 1114, which is a cyclin D1, and which is associated with a good prognosis, the 414, which is the MMZ translocation, which is associated with poor prognosis, and 1416 and the 1420 translocations, which are also having a poor prognosis. And we still, although we have made a lot of progress in the treatment of myeloma, it's especially in those poor translocations that we still have quite a bit of problems getting the disease under control for long periods of time. Getting disease under control is not that difficult, but keeping it under control for a long period of time, that's a totally different issue.
Okay, and these are just features where certain genes are, so we have these features separated out in these Facebook groups. And when different therapies are introduced for a specific kind. For example, deletion 17 patients might be better off with bortezomib or something like that. Then you can start to get to a more personalized therapy, and you're finding this out, these genetic features through the FISH and the gene expression profile tests, or what other testing are you doing to find these translocations?
No, that's exactly right, through the gene expression profiling and through the FISH analysis. And
okay. So when you go, Dr. Zhan, maybe you can explain the process that you went through to find or to discover and what you would like to talk about today, because I think the process that you're going through is really, really interesting.
That's right. Yeah. We have developed and validated 7 genetic subtypes, such as c-MAF and MAFB, cyclin D1 and cyclin D3, and MMC activating translocations, and also hyperdiploidy using gene expression profiling. So those indicators of this deregulation of common pathways by gene orthologs, common gene signatures were observed in cases with c-MAF and MAFB. That's why we classify the c-MAF and MAFB as the same subgroup, but they have a different genetic translocation. c-MAF is come from 1416, and MAFB due to the translocation 1420. Then we using those 100 genes uniquely dysregulated in the MAF, including both c-MAF and the MAFB subgroup. We predict 5 compounds that could induce or reverse desired gene expression changes based on the connective map. This database, Connect database, is comprehensive, including compounds and the microarray dataset. However, we only found the drug named Alsterpaullon significantly inhibit myeloma cell growth with extremely high expression either c-MAF or MAFB. That's the reason we came to our proposal and defined this Alsterpaullon as a drug to target this specific subtype of myeloma disease. In addition, we also found that expression Notch2 is much stronger regulated in this MAF subgroup than other subtype of myeloma disease. And the Notch2 is an important gene related to cancer stem cells, drug resistance, and also microenvironment. So in another drug, we're trying to bring in targeting this subgroup is Notch2 antibody. So that's the whole process for our project.
Okay, that's a great summary. And I'm going to ask you to kind of dig a little deeper, or this is pretty complex stuff, so I want to make sure that patients understand it. So your first step was that you divided all your samples into 7 different genetic subtypes. And each of those different subtypes deregulate different genetic pathways. So then you were focusing on just the 1416 and the 1420 translocations.
That's right.
And those affect something called a gene called c-MAF for the 1416 and MAFB for the 1420, right?
That's right. That's correct.
Okay, and then your second step was you tried to look at all the genes that were deregulated for those 2, and you found 100 genes, right?
Yes, we used the top 100 significant genes. The gene is more than 100 expression in this subgroup, but for this query study to the database, we only chose the top 100 genes for this analysis.
Okay, so you're looking through a massive database of genes, and you're trying to find the genes that might be causing these problems. Is that
That's correct?
Just
yes,
a very
exactly.
basic. Sorry, my way of saying it is very basic. And then you connected that, what you found with those genes, to existing drugs or could be drugs, to then try to match up a drug with those mistakes in the gene, right?
Yes, so we used the 100 uniquely dysregulated gene in this translocation of 1416 and 1420 into the connective MAP, c-MAF database. In this database, it includes 1,200 compounds and a few thousand microarray. So once we input our 100 unique genes, we can, based on the score for connectivity or enrichment, we can find which compound can reverse this MAF signaling pathway. That's the strategy we're using. Then finally we found 5 compounds can do that function. But based on the bench work, we finally only found 1 compound can inhibit MAF signaling pathway and kill myeloma cells.
Okay, so your next step was that you matched that compound database up against those genes, and then you found 5 compounds that potentially could, but then you only found really one compound that could reverse that bad signaling pathway, right?
Exactly.
Okay, and that is called Alsterpaullon. How do you say that? I don't know how to say that word.
Yeah, Alsterpaullon.
It's spelled Alsterpaullone.
And it seems like it's also called, they just mark it as ALP, right?
That's right, yeah.
Yes.
That may be easier.
To make it easier, yes.
So can you explain, either one of you, what ALP does and how it changes that? Because when I was reading through your proposal, you mentioned that these two, the 1416 and the 1420, that are c-MAF and MAFB, they're resistant to bortezomib. And since most of us are getting bortezomib as part of our treatment, can you explain what that gene is doing and then how this drug is affecting it?
Okay, I go first. So ALP is a small molecular compound which belongs to the class of organic compounds known as benzodiazepines. These are organic compounds containing a benzene ring fused to azepine ring. So that's the structure for this compound. And the function for this ALP is an inhibitor for cyclindependent kinase and also Wnt signaling pathway inhibitor. That's already known. As we also know, the translocation 1416 and 1420, the both pathway about cyclin-dependent cell cycling pathway and the Wnt signaling are activated too. So that's good evidence for using this drug to target this pathway. Then about the resistance to Velcade, bortezomib, we already know, actually not done by our group, from the published data. One group report that bortezomib can phosphorylate either MAF or MAFB. The good thing is if phosphorylate MAF or MAFB can degrade these 2 oncogenes or NAST genes. In another aspect, this bortezomib can also activate MAF or MAFB downstream signaling. In my expectation, it seems the later one plays a major role. That may be the reason bortezomib also activate MAF or MAFB downstream signaling. That's why maybe it's resistant to bortezomib.
So you're saying bortezomib creates sorry, I'm not understanding it as well as I should. Well, it's degrading the active signaling?
You would expect that bortezomib would prevent the downstream signaling of MAFB. MAFB if that signals, it does bad things. And you would like to have a drug that prevents these bad things from happening. But actually, when Frank did his analysis, what he saw is instead of inhibiting this downstream signaling of MAFB and all the bad things, it actually, in some ways, activated this downstream signaling. And that's why it didn't work. It doesn't work very well. It works for a while, but then it stops working.
Interesting.
Right.
Yeah. One of the basic problems in myeloma is that there are so many redundant pathways. You block one pathway, it fires through another pathway. And you think you're doing well, but actually the only thing you're doing is, yes, you block one specific pathway, but now it starts to fire through other pathways, which will lead to the same end result, meaning resistance to the drug.
So you need not just one blocker. You need a whole team. You need a whole team of blockers.
Yeah, there is only one disease in hematologic malignancies which can do with a single blocker, and that's chronic myeloid leukemia, because everything has to go through the Philadelphia chromosome. But in all the other diseases, it is much more complex, and a single drug has never worked in any of those diseases.
So what would ALP do in with the MAF and the MAFB?
ALP is a cyclin inhibitor. That means that the cyclins are necessary for the cells to grow and proliferate. And if you inhibit that, then the cells cannot grow and proliferate anymore. Okay.
And then you wouldn't get, would you still be able to use it with bortezomib?
That's one of the things we would like to test, whether if we combine this Alsterpaullon ALP with a bortezomib, whether we can make the cells sensitive again to bortezomib. But we don't know that yet, but that's one of the things we absolutely want to see.
And have you seen the same effect in carfilzomib that you see with bortezomib in terms of that it's activating it in a negative way?
Yes, they're all of the same class of drugs, so the proteasome inhibitors, and they all work the same way.
Okay, so no matter what proteasome inhibitor you use, whether it's bortezomib or carfilzomib, it would have the same outcome.
Yes.
Oh well, that's not good, because that's like a standard therapy that's used in most everyone's treatment.
Yes.
Okay, so when it affects, it inhibits proliferation, and then it allows the other drugs to work. And then you also, Dr. Zhan, you also mentioned that there was another aspect to this, that you found a Notch2 pathway. Can you both elaborate on that?
Yes, I go first. Dr. Jacob may after me. Yes, Notch signaling pathway is important signaling in normal hematopoietic stem cells, embryonic stem cells, and cancer stem cells. So this pathway is widely studied in other cancers. Even in myeloma, we already reported that if we block Notch signaling using gamma secretase, can induce myeloma cell apoptosis and kill myeloma cells. However, that drug is not specific to target Notch2. In our proposal, we found Notch2 is one of the Notch receptors. So Notch receptor include Notch 1, 2, 3, and 4. So this Notch2 is uniquely upregulated in this MAF subgroup of myeloma. Our idea is if we can use this Notch2 specific inhibitor or antibody, then we can personalize to treat these spatial translocations. And also, currently the Notch2 antibody is available in clinical trial. Widely is in the phase 1 and 2 for pancreatic cancer, lung cancer, and other solid tumors. So that's why I like to bring this NOTCH2 antibody in targeting this unique subtype of aggressive myeloma disease in this proposal.
Okay, so NOTCH2 is a signaling pathway, and you found this. What's the difference between an antibody versus an inhibitor?
Well, yeah, there are many ways to deal with a receptor. One is you can use a small molecule that inhibits the function, or you can use an antibody that blocks the function. Ultimately, the end result is about the same. But in general, if your receptor is not mutated, if there are no changes in the receptor, you're better off with an antibody than with a small molecule inhibitor. And then there is another group of drugs which he talked about, the gamma secretase inhibitors, which are not selective. And you have to remember that these pathways are not only important in cancer, but they're also important in normal blood formation cells and in the gut cells that we make every day, and normal stem cells that we have in our body everywhere. So the more selective you can be, and the more you can only inhibit what you really want to inhibit, and not inhibit things that can only be detrimental and have no benefit to myeloma, the better off you're going to be. That's why this antibody, which is very selective for NOTCH2, will be a better approach than generally inhibiting NOTCH as a whole.
Because it sounds like there's NOTCH1, NOTCH2, NOTCH3, NOTCH4, and you might stop something that you don't want to stop.
Exactly. You can imagine that if you block all NOTCH, that you will have problems with your normal blood formation, the normal hematopoiesis, and also the normal cells of the gut, the stem cells of the gut. And you see that if you give those gamma secretase inhibitors, one of the common side effects is that these patients have a lot of diarrhea because you mess around with their gut stem cells.
Interesting. And that, I think, is the challenge, I think, of multiple myeloma, because there's not one single target that you're going after. You're going after all these targets. So in trying to find drugs that are effective, you can either shut everything down, which would be dangerous,
yes,
or you could try to be very specific, which is what you're trying to do in this proposal. You're trying to say, for these patients that have this specific translocation, this might be their silver bullet, or their target that might work for them specifically.
Yeah, there is something in cancer that they call oncogenic addiction. That means that cancer cells become addicted to a certain oncogene. And if you can block that oncogene that makes the cells survive and grow and do well, you can basically prevent this from growing. While the normal cells, they don't have this addiction. They depend on many different pathways, and they cannot be inhibited by just a single intervention or a few interventions.
So you're trying to isolate those pathways that are most effective only for the cancerous cells.
Yeah. What we want to do is try to isolate the pathways which are majorly upregulated in the 1416 and 1420, much more than in normal cells. So the more they are upregulated, and the more they are different from normal cells, the better we like it. That's how we can selectively go after the cancer cells.
And that's what flags it for you.
Yeah.
That's. Okay. And can you explain what your next step is then? If you've gone through the database, you found the compound, and now you're going to try it in multiple myeloma. Can you explain what happens next?
Yes. So in our preliminary data, we tested this compound in myeloma cell lines. So these cell lines including either with translocation 1416 or 1420, or without these translocations. Then the ILP we found this drug can uniquely kill those myeloma cells with translocation, but low effective in those without 14-16 translocation in this myeloma cell lines. So after that, in our proposal, we try to do some in vitro and in vivo study using primary myeloma sample, because this is the key for translating this drug into clinic. So we propose to at least collect 5 myeloma sample with or without this translocation, then treat this using this drug to do in vitro co-culture myeloma cell with stroma cells within this in vitro model. Then we will also do the SCID U mouse model that exactly mimic human myeloma disease. We inject this tumor cell into fetal bone, human fetal bone, into the mice. So from this case, we can get the idea if this drug is still specifically targeting those translocations in primary myeloma sample. That's the first step we're doing for ALP, but we are also test the Notch2 antibody for this specific translocation samples.
So are
also,
you?
Yeah.
Go ahead.
Maybe we also do combination with these 2 drugs or with others such as bortezomib or IMID, other drugs that we try to propose in the first aim. So another one we're trying to do, even we know some targets from c-MAF or MAFB, but it still cannot clear what the direct targets regulated by these 2 oncogenes. So we try to use the new technology because the next generation sequence just it's a really powerful tool, can be used to identify c-MAF or MAFB direct targets. If that's the case, if we found some novel targets, and we can also see if any drug can target those targets, then can be easily translated to clinic again. So that's our second aim trying to do in this proposal.
Okay, well, let me ask you a question about these different compounds. I know these compounds are in this database and you're matching them up. I'm just out of curiosity, who's developing these compounds, and then, because you're testing samples, and it sounds like your first step is to do it in the lab in vitro in a dish, and then try it in actual live mouse models as a second step, and then maybe with or without bortezomib, and with or without this Notch2 inhibitor. But I'm just curious where these compounds come from. You said it was an organic compound, but I just think it's amazing that these compounds exist in a database somewhere that you can use to test.
So actually, we bought this ALP compound from Sigma company. We're trying to contact this Notch2 antibody from Onco-made pharmaceutical company because they provide this compound for clinical trial. Dr. Checo is trying to call them this week or next week. So that's the plan. We're trying to get contract with them, then we can test in our model.
Okay, and this other drug that's the Notch2 signaling pathway. What's the name of that?
It's named Terectamab.
Terectamab. And you said it was being used in phase 1 and phase 2 trials for pancreatic and lung cancer already, correct?
Yes,
exactly. That's correct.
Yeah. You have to remember that when the drug companies develop a drug, that they are always trying to go for the large markets.
Mm-hmm.
And so compared to other solid tumors like lung cancer and breast cancer and prostate cancer, myeloma is a relatively small fish. So they will always try to see whether their drugs work initially in tumors that are much more frequent than myeloma.
Mm-hmm. And then I think you layer on top of that, a small percentage of myeloma patients have the 14/16 or 14/20 translocation.
About 6% of all myeloma patients have this translocation. But those are actually the patients that are doing the worst. when we look at our overall profile, about 83% of our patients are doing very well with the current treatments, and about 17% are not doing so well, and actually are not doing too much better than 10 or 15 years ago. And it's especially those with these translocations, the 414 and the 1416 and 1420 translocations, and the deletion 17q. Those are the patients that we still need to do a lot better and make sure that their prognosis becomes as good as what we see in other patients.
Well, and I think that's one of the reasons why this MCRI project exists, is because the funding might not come from the large pharma companies to go after a small target, but if you can find a cure for multiple myeloma in one type, then it might not be the same as other translocations or other genetic features, but you can say that you've solved one piece of the puzzle.
Yes, and then again, there will be other cancers that have the same 100 genes that are differentially expressed compared to others, and in those it should also work.
And I have a question about that also, because you look at some patients, like I know that Mike Katz just recently passed away, and he had multiple myeloma, and he also had colon cancer. Do the same genetic deregulation of the genetic pathways exist in your mind for both types of cancers? Like, could he have had the same genetic errors, I guess?
There is no direct evidence that the same important genes for myeloma will also affect colon cancer, but it's not sure that when you would look at the cancer stem cells in colon cancer and in myeloma, that those genes may be much more comparable than if you look at the bulk of cells. So what we typically do is we take a sample of a patient with myeloma who is just diagnosed, and the bone marrow is almost full of myeloma cells, and it's very easy to find the gene expression profile. But we also know that about 80% of the patients go into complete remission with appropriate treatments. And that means that in 80% of the patients, you eradicate more than 99.9% of all the myeloma cells in the body, meaning that those patients have very sensitive cells to chemotherapy, and that there's only a small percentage of the cells that is much more drug resistant. And we, instead of looking at the gene expression profiles in the beginning, like most people do, we have been always more interested in looking at the gene expression profiles of cells that have survived our intensive treatments and by definition are drug resistant. And we use our chemotherapies as a way to select for the most drug resistant cells. And we think that that is a much more informative approach than to look at what the gene expression profile shows at the time of diagnosis.
Because you're just determining what's surviving the chemo. Exactly.
What are the cells that are surviving, and what do the genes show us? And is there something in common between all those patients that have cells surviving the treatments? And if there is something in common, how can we attack that and make those cells go away?
Okay.
And our research has basically shown that the major problem in myeloma is the myeloma stem cells, which are the mother cells. We used to think that in cancer, every cancer cell is about the same and can cause the disease to relapse and to grow. We now know that there is only a very small fraction of cancer cells that can do that, and those are the cancer stem cells or the mother cells. While the daughter cells, they have a limited lifespan, and after a while they will just die, and they cannot renew it, they cannot replenish themselves. It's only the stem cells that can do that.
Okay, so you're trying to target the source, basically.
Exactly, and that's why we are so interested in Notch, because Notch is a typical gene that is upregulated in all types of stem cells, whether it's cancer stem cells or hematopoietic stem cells or embryonic stem cells. It's upregulated in all those stem cells. But we want to go for the notch that is the most relevant to myeloma and not relevant to other diseases and other normal cells.
And how do you test for an upregulation of Notch2? What test shows that?
Well, you look at the amount of protein of Notch2 in the myeloma cells that have the 14-16 translocation or 14-20, and then also in the ones that don't have those. And you see much more Notch2 protein in those cells that have the 14-16 and 14-20 translocations. And there are guys in the Netherlands who have taken myeloma cell lines that had very low expression of Notch2, and they introduced the MAF gene, so that the MAF gene was overexpressed in those cells. Although originally it was low, now after transfection there was high expression of the MAF genes. And what they saw is as soon as they introduced that MAF gene, Notch2 goes way up.
So that was a clear result.
Yes. And then Dr. Zhan has also shown that if you then take the opposite, you have cells that have high expression of MAFB, and you block Notch2 in those cells because the high MAFB have always high Notch, that those cells are dying. He has shown that in vitro in cells.
Yeah, originally I discovered this Notch2 based on our gene expression profiling from almost 500 samples. So in this specific subgroup of c-MAF and MAFB, we found Notch2 is at least twofold higher than other subtype of myeloma subgroup. That's why it attracted us to target this molecule in this aggressive subtype.
Yeah, and again, remember that the more the abnormal cells are different from normal cells, the better we like it. And the better you can specifically target, and the more differential effect you will see on the cancer cells versus the normal cells.
Just because they're more specifically defined?
Well, they have higher expression of Notch2 than normal cells. And so if you block Notch2, they will be much more affected by that than in cells that have also Notch2 but at a much lower level.
Okay, great. Do a lot of patients that have 14-16 or 14-20 have multiple genetic, genetic issues? Because sometimes I know that for deletion 17, patients will pick up that deletion over time. Is that the same with 14-16 and 14-20, or do you have it and then just keep it?
No, you don't have it. If you don't have it, you never get it. If you have it from the beginning, in contrast to deletion 17, which you can acquire.
Right. Okay. Okay, perfect. So, Gary and Jack, do you have any questions? I have several more, but I want to allow you time as well.
So this is Jack. I have several questions, but boy, this has really been educational. I really appreciate the doctors trying to dumb it down for me.
Yeah, same for me.
Thank you.
So, just to clarify for my sake, I think I've read that T14 and translocation 14-20, 14-16, and 14-20 each occur in about 1% of myeloma patients. Is that true?
That's underestimating it. If in our studies, we found that 6% of the patients have either one of those translocations.
So that's about 3% for each.
Yes.
Okay.
But 1% of each is an underestimation.
Right. So 3% or so each. Okay. And do all of those T14, 16, and 14-20s have this oncogene c-MAF or MAFB?
Yes.
Yes.
And do those oncogenes get expressed? And you might have answered this, I apologize. Do they get expressed in other normal cells as well?
MAFB is expressed in other cells too, but at a much lower level.
So hopefully that giving ALP, for example, which inhibits their expression, won't have significant undesirable side effects?
No, when Dr. Zhan looked at that, when he looked at the cell lines that had the MAF translocations, those were killed. But all the other myeloma cell lines that didn't have the MAF translocations, those were unaffected. So we also think that the normal cells are going to be totally unaffected by this.
And also those translocations, we name the spike expression of c-MAF and MAFB, means those expression is more than 10,000 higher level than those without translocations. That's why we also name that may be a gene addiction or oncogenic addiction, this specific oncogene. In our preliminary data, as Dr. Treacle mentioned, our drug LP only kill this myeloma cell with translocation 1416 and 1420, but not on those without translocations. So our prediction is the side effect should be very, very low in normal cells.
What about in other myeloma cells? Do those c-MAF and MAFB oncogenes appear that don't have a translocations?
Right. Just so the one report from NIH a few years ago, this 414 translocation activate the MMSC actually can upregulation c-MAF, but that upregulation is much lower than this original translocation by 1416 or 1420 translocations. That's also more than at least 100-fold difference.
And again, this comes back to what I was trying to explain in the beginning, that if you have this translocation, you now bring this gene under a different promoter, a much stronger promoter, a much better igniter to get this gene going than where it is originally. And if you don't have the translocation, you can still have increased levels of c-MAF, but at a completely different level than when you have this translocation. And again, the difference is about 10,000 times.
Got it. I understand. Those are my questions, and I really appreciate it. I'm learning as you are discussing it, and it's really informative.
Thank you.
And I have one more, before Pat asks his questions, I have one more follow-up based on what you just said. So can patients have the 1416 or 14 translocation on some of their myeloma cells, but not on all, or do they have it on all cells? So if you go after it with this target, it would all be killed, or would you need some kind of companion thing to go after, let's say, like this hits 50%, and anyway, does that make sense?
All the cells in patients who have the c-MAF translocation, all the myeloma cells have this translocation. But your second question, do you think that you can kill all the myeloma cells with a single agent? I think that's quite naive. I think we need to have combinations. That's why we are trying to combine this Alsterpaullon with the Notch2 inhibitor and probably with other drugs to see whether these combinations can kill all the cells. But a single agent killing all the cells, I think that doesn't happen.
Just because there are too many ways of myeloma getting around it.
Exactly. There's always too many ways to fire through other pathways and getting around the block. You put a block up, and they will go around it and still get to the same end point.
Okay, perfect. Okay, Pat, did you have questions?
I do. Thanks, Jenny. And can you? Is there anyone better at explaining this technical material than Dr. Treacle?
I think it's great.
Doctor, you're the best. So this was a good lead-in for my question, because I wondered, first of all, it's great just imagine you're one of these patients and you know that Velcade or Kyprolis isn't going to work, and so why even give it to them? And just knowing that is a service to a patient's quality of life. But like Jenny said, unfortunately, that takes a whole class of drugs out of the mix. So you get this figured out, and you successfully attack these particular, this is working. So I guess the $1 million question is, how long does it work? I mean, what happens in the 3 or 4 or 5 years? Does it relapse just like someone responding to IMiDs and then the IMiDs stop working?
Obviously, without doing a clinical study, we cannot answer this. But before we can do a clinical study, we need to have more convincing data that this works indeed not only in the tube, but also in the mouse model, where you have the normal microenvironment of a human system, where a lot of our drug resistance is caused by the microenvironment, by the soil where the myeloma cells grow in. And if you don't take care of the soil, you cannot get the disease eradicated. So we want to do this also in vivo, in mouse models, where you have a normal human fetal bone implanted with normal human microenvironment. You inject a patient's myeloma cells in there, and then you give the drugs to the mice, and you see whether you can kill this myeloma. And when we can do that, then we are ready for clinical trials.
That's great. So in your experience, when you do that, how well does the mouse model really translate to the clinical trial? I know patients get excited in vitro, they get excited to the about the preclinical stuff, and then there's disappointment or trials never even get off the ground. So in your experience, how often does this work? So things are working as you expect in mice models. You guys are incredibly smart guys. Let's assume that's the case. So what are the odds that it'll translate to humans and this thing might actually work?
That probability is not higher than about 10%.
Really?
Yes. We have cured myeloma in the mice 100 times over, but we have not cured myeloma in the large majority of patients with myeloma. We can make them live long. There are some with good prognosis that live longer than 15 years and are probably cured, but that's still a minority. The proof is always in treating patients and see how long it will take for the disease to come back.
Sure. What an incredibly honest and yet disconcerting, disappointing answer. But,
wow.
But 10% is still, you guys, come on. I can't believe it's only 10% for you guys, but thanks for being so modest and honest. But this sounds like, it sure sounds like you're on the right track.
Yeah, when I say 10%, I base that on it's 10% of all the compounds that are developed by the pharmaceutical companies,
sure,
and that have done all these same studies in the test tube and then subsequently in the mice, and only one out of 10 products that they think looks very promising is actually, when they do the human studies, very promising.
Gotcha. And you're probably just like patients. You're always expecting to be in the upper half or the upper 10%, right? So you wouldn't be doing this if you didn't think it's going to work, just like a patient wouldn't try a certain therapy working with their doctor, hoping and expecting that it wasn't going to work for them. But we'll keep our fingers crossed. And
we all hope that we live on Lake Wobegon, where the average child is, where every child is more than average.
Thanks a lot, Doctor. Thank you, Jenny.
Thank you.
Okay, thank you so much, Pat, for your questions. Well, a couple of questions that I have. If you have the 1416 or 1420, and would you delay getting bortezomib or some kind of proteasome inhibitor, or would you just add that to the mix right away?
No. The reality is that patients with 1416 and 1420 translocation have the same chance of going into complete remission as patients who don't have this translocation. The difference is that in the other patients, the responses last for long periods of time. In these patients, they tend to be short-lived. But you can still debulk and get the patients to very low doses of tumor cells with those drugs. But after you have done that, then you need to come in with these drugs that are specifically targeting the specific translocations. This is not like you would think I'm going to give this to newly diagnosed patients with 1 trillion myeloma cells. You first want to debulk with what you know works, and when you have only about 1 million to 10 million cells left, you want to kill those with those specific drugs.
Because they might not have to be that strong in order for you to actually target. And I think one of the reasons that 1 in 10 might work, that are these compounds that are tested, is because I think previously there wasn't a lot of very gene-specific testing being done.
That's true. Absolutely true.
And so you weren't separating or segmenting and looking through this database to say, okay, we just want to look at the c-MAF or MAFB gene to target that. We're going to look at myeloma in general, and we're going to try to screen for, thousands of drugs or something that might work. But since myeloma is all different, then how do you know?
Yes, exactly. And then again, if you would take this drug ALP and you would give this to 100 myeloma patients, and you would see a response in 1 or 2 patients, you would say that's not a very effective drug. But the ones that are responding were all the ones that had the MAFB or the c-MAF translocation, and you missed that if you didn't do the gene array or the FISH or whatever.
Right. And so that's the bonus of the new gene expression or the new genetic testing and the focus on that.
Yeah, and we think that basing therapy on gene expression profiles is a better way than has been done in the past. But again, we don't have hard evidence that's the case, but we think that that's the case.
And I guess I would stress to patients that if I attended a patient seminar, this was 4 years ago when I was diagnosed, and asked all the patients to raise their hand if they knew their genetic features. I would say less than 10% of them raised their hand, which I thought was crazy because why wouldn't you want to know what you're dealing with? So I just really think it's important for newly diagnosed patients especially to know what they have, and that comes with very thorough testing, including the genetic testing.
Yeah, let me emphasize again that in myeloma patients there are 2 things that are important. One is what do their genes show, what are the genetic features, and number 2, how extensive is the disease. And those are basically the only 2 real things that matter in myeloma. How much disease do I have, and what are the features of my disease?
Because that helps you craft a plan, right?
Exactly, yes.
Okay, my final question will be just expected milestones. We already talked a little bit about what you would like to do next, that you'd like to study it in, in vitro and then in mouse models. But could you give us an idea of the milestones, how long that might take, and an estimated cost in general, a ballpark?
Right, yeah. So we propose for 2 years grant. So the first year, I think we can do on this myeloma cell line study, test this drug. But we need a little bit more time to collect enough primary myeloma sample. That's why we need 2 years for this proposal. Then for this new technology thing, we can finish the cell line experiments in the first year. But for clinical sample, get enough sample of cells, we need 2 years too. So the estimate. Then I hope we don't have detailed budget in this moment, but we hope we can apply about $300,000 to $500,000 for this proposal.
Okay, great. Well, we've been asking that question to everyone just to get an idea of how long the project would take and when it could be seen potentially in the clinic. So that's very helpful.
Yeah. So let me make sure that we all understand that the 2 aims of this project are, number 1, does the 2 drugs that we have discussed, do they work in the mice and in the test tube the way we think they work, and do they work better in combination? And the second aim is can we find other important genes in MAF patients that we can also use as targets so that we can even do better than with the 2 drugs we have. So those are the 2
great.
aims. Yeah.
Okay, perfect. And Gary actually had a follow-up question that was a writing question that he sent. He said, with gene deletion 17 seeming to be the largest genetic high-risk features, is there the same kind of analysis that you've gone through using this genetic database that can be conducted for this deletion as well?
The 17 is a more difficult thing to do because, in contrast to MAFB and c-MAF and MMZ, those translocations are present in 100% of the cells. The deletion 17 is often present in a fraction of the cells, which can go from 14% to 100%. So if you try to collect your myeloma cells, you have no idea how many will have the deletion, how many will not have the deletion. And the gene expression profile becomes much more difficult to do because it's often an acquired abnormality.
Yes, we published a paper in 2008, Blood. We classified, we also identified the relative signaling pathway for P53 deletion. That's totally different from we saw in this translocation 1416 or 1420. As Dr. Treco mentioned, the signal or the downstream targets not so unique in the P53 deleted samples.
Okay, great. Well, Drs. Zhan and Treco, we are so happy to have you on the show. You've described something that is so specific that patients with these translocations have got to be just jumping up and down that people are looking at this for them on their behalf. I think it's remarkable that we could target something so specifically and really help people that are struggling the most, I would say, really find something that could completely change the course of their future.
That's what we are hoping for, and I hope we can deliver. But we will certainly try as hard as we can.
That's right.
Well, thank you very much for joining us today, and we just really appreciate your participation in this project.
Thank you all.
Thank you.
Thank you, gentlemen.
Thank you for listening to the Myeloma Crowd Radio Show and the new MCRI series. We believe patients can help support the discovery of a cure, and we encourage you all to become involved.
Hello, it is Ryan, and we could all use an extra bright spot in our day, couldn't we? Just to make up for things like sitting in traffic, doing the dishes, counting your steps, you know, all the mundane stuff. That is why I'm such a big fan of Chumba Casino. Chumba Casino has all your favorite social casino-style games that you can play for free anytime, anywhere with daily bonuses. That should brighten your day a little. Actually, a lot. So sign up now at ChumbaCasino.com. That's ChumbaCasino.com.
No purchase necessary. VGW Group. Void where prohibited by law. See terms and conditions. 18+.